Hello everyone, welcome to a lab science flow cytometry classroom. During the flow cytometry experiment, we need to prepare the test sample. Meanwhile, different control groups also need to be set up to help us obtain more accurate and clear experimental results. In a flow cytometry experiment, how many control groups are needed? In these videos, we will summarize 5 common flow cytometry control settings for you, providing a reference for sample tube settings. Today, I will introduce the introduction and functions of blank control and single positive control. First, let me introduce the blank control group. Blank control is a control tube without any fluorochrome dyes added. A blank control is set up to distinguish the autofluorescence inherent in the cells and to determine the autofluorescence introduced by external factors such as drug treatment, thereby eliminating the influence of autofluorescence on the experimental results. The position of blank tube cells in the flow cytometry experiment result graph is the location of the possible total negative cell population from crossing the line due to too low voltage when starting up the machine. Next, let's talk about the single positive sample group. The single positive control, also known as the single positive tube, is a sample tube with only one type of fluorescent labeled antibody added. In multicolor flow cytometry experiments, due to the wide emission wavelength coverage of fluorochrome may overlap, causing certain interference to the data analysis of the experimental results. In this case, we usually set up a single positive tube for compensation and adjustment. If there are 3 types of fluorescence in the experimental panel, 3 single positive tubes need to be set up. During the detection, the results of the experimental group can be compensated and adjusted based on the detection results of the single positive tube to eliminate the interference caused by fluorescence overflow, thereby obtaining more accurate experimental results. The single positive tube can also assist us in adjusting the channel voltage to prevent the signal from exceeding the receiving range. The introduction of the blank control and the single positive control comes to an end here. The subsequent videos will also introduce experimental groups such as isotype control and FMO control to everyone. That's a wrap for this video. Stay tuned for elaptions, and let's keep pushing the boundaries of discovery together.